Well, here's some quarantine excitement. I'm gonna make one of these videos in a new location in my house. So let's meet the cats who never come downstairs. There's Marmy. Here's Eileen, who has 3 legs. And here's Rosie, who has 1 eye. And if we head in here, this is my bedroom. And I basically wanted to show off my DNA carpet, which, you know, huge surprise. And finally, a big shout out from Peg, the 3-legged cat, who's taking it easy. Excitement, I know, the view. All right, so let's finish up PowerPoint number 1 and head on to PowerPoint number 2. Okay, so here's where we left off. Told you about, here, I'm gonna try to make this a little more stable. Let's see if I can do it. I'm sorry, I'm a boomer. Sorry. Okay, yep, that sucks. All right, I'm just gonna have to try to hold it steady. I'm so sorry. Thank you for your patience. So we left off talking about the X and Y chromosomes. And remember, if a person has 2 X chromosomes, they are a genetic female. If they have an X chromosome and a Y chromosome, they are a genetic male. All right, so here's where we left off. So as I mentioned, the main type of forensic DNA typing is nuclear DNA. So it's DNA from the nucleus and looking at STRs, or short tandem repeats. So each individual, each different human, is going to have different numbers of repeats on each of the 2 chromosomes that they have. Because remember, chromosomes come in pairs. You get 1 from mom and 1 from dad. So, God, I'm sorry about this. So, for example, let's say we type an STR, a forensic marker that's present on chromosome pair number 1. So, if you received 4 repeats, remember I'm thinking of these as boxcars, on 1 chromosome, and 9 repeats on the other chromosome, and we can't tell which one is coming from mom and which one's coming from dad, but your STR type for the location, the forensic marker on chromosome 1 would be a 4-9. And so the way that these types are displayed, they're always 2 numbers and there's a comma in between them. So here is an example of the different markers. And I know this can be a little overwhelming, but all I want you to see from this is that when we look at markers, forensic markers, and they have big fancy names, you don't have to worry about those. You certainly do not have to memorize where on what chromosomes they are. What I want you to see is that the vast majority of them, except for D5 and CSF1PO, they're all on different chromosomes. And that is done for a reason. So if you can flash back to high school biology, one thing that happens during the process of the formation of sperm and egg, which is the process of meiosis, is that the chromosomes are independent of each other in terms of how they line up and which chromosomes end up in sperm and egg. And so for forensic typing, if we put the chromosome markers on different chromosomes, or in this case, really far apart on the same chromosome, they are independent events. So if you can remember way back to when we talked about probability and how if you have independent events, like a coin toss, you can multiply the probabilities together. That is where we're getting these huge, very discriminating numbers. So basically, each forensic marker is independent of every other forensic marker, and they're all like independent coin flips. So the chromosome, forensic DNA type that you have on chromosome 8 has no bearing whatsoever to the forensic DNA marker that you have on chromosome number 2, and etc. The last thing I want to point out is this is called amylogenin, and that's where we get our gender marker. So that will tell you whether a person is, whether the sample originated from a male or a female. Okay, so here's what's important to summarize. Because each chromosome pair is inherited independently of every other chromosome pair, DNA types on separate chromosomes are considered independent events. That means the probabilities of DNA types on separate chromosomes can be multiplied together. So in STR typing, we look at 13 different independent locations on the probability of a match. And so often that probability is in the billions, trillions, quadrillions. I've seen, match probabilities up to the quintillions. And all of those probabilities usually far exceed the population of the Earth, which is currently about 8 billion people. So when you see those probabilities in the, billions, trillions, quadrillions, what that basically means is that this DNA profile that matches the defendant or the suspect couldn't come from any other person on the planet Earth unless they have a same egg, twin or triplet, a same egg sibling. So, and once again, also STR typing can tell you the gender of the person. So here is some sample STR data. So the word locus is the scientific word that just means the location on the chromosome. You can think of it as like the house address on a particular street. And so we see that on the evidence, for example, the chromosome marker in the top row, which is on chromosome 3, it's called D3. The evidence sample has a 1517. And when we compare it to suspect A and suspect B also has a 1517. And so that is a match. And then you basically do this for all of the different locations. And we can see that suspect B, if you look up and down the columns, matches at every single location. That means that you can take all of those frequencies that are listed in the right-hand column and multiply them all together, and you're going to come up with a very, very small number in terms of the number of people on the Earth who could carry that particular profile. Okay, so I just wanted to show you, this is called a chromatogram, and this is actually what the actual DNA profile looks like. So it kind of looks like chemistry data, but you can see it has the different locations, and then these little peaks correspond to the numbers. So I just wanted to show you what DNA, what the actual DNA analysts are looking at when they are interpreting data. Okay, so today I'm going to take a shower in honor of you guys. I'm going to get this uploading. So we're done with PowerPoint number 1. Also, be sure to check. I just uploaded a DNA base pairing activity for you guys to do. That is due on Friday. So please check that out as well. We'll start PowerPoint number 2 as soon as I can. All right. Thanks. Bye.